So let's then move on to the computational part here. And just as for experimental techniques, it's a very broad and diverse field, and it would be sort of like impossible for me to give you a fully comprehensive overview of everything. So I will instead go into certain categories that I think are fairly well established and common to work with. So we have the single cell inspired method, spatially aware methods, deep learning, and integration with single cell data. Those are the four that I will dive a bit deeper into. And these, I should say, are my own categorization scheme, sort of. So don't take it as like the law, but I think it's a nice way to structure these different methods in my brain, at least. So to start with the single cell inspired methods, the basic idea here is that you try to apply already existing methods and tools developed for single cell data, but perhaps to your spatial data, but perhaps you fine tune them or make them more suitable for your specific spatial data. And examples of this could be to cluster your spatial data and then show these clusters in space. You could try to decompose the expression profiles using certain factor models, or such as LDA or use standard factor analysis. You can try to do trajectory inference, and there are different ways of doing this. Perhaps you treat, again, your spatial data completely as single cell data, or you try to reconstruct or repurpose the already existing algorithms. And there are several different tools that provide tools of doing this. CRAT, for example, has added support for spatial data, not every kind of spatial data, but for example, Visium and even ST fits in that. ScanPy has also added support for spatial data. And of course, these suits do not only offer single cell inspired methods, but as they were mainly developed for single cell data or even bulk data, a lot of these methods can then be applied to the spatial data just straight off. We also have ST Utility, which is built on top of CRAT, but very tailored for spatial data. I should actually say here, tailored for Visium or ST data. It was developed by two people in our group, since they work so extensively with just ST and Visium data. Then we have ST Learn, which is built up on ScanPy rather than CRAT, and again tailored for certain forms of spatial data. Also very recently, and with that I mean yesterday, some people submitted a manuscript to bioRxiv where they presented this new library called Spatial Experiment, which is supposed to be very similar to the single cell experiment library and ecosystem in R. I have not had the time to check that out, but yeah, at least it exists. And there are a lot of more suits and tools coming and already existing for spatial data. But I wanted to give some examples here now what I mean with this clustering and trajectory inference. So here we see an example of some results that we obtained in a study that we were doing on human breast cancer. And to the left, in the very leftmost picture, after doing a very similar process to what you would do with single cell data, you normalize, you cluster, and you project your data or embed it in two-dimensional space using UMAP. You can get this neat embedding here, where you can color each of your data points by the cluster identity. What is then pretty cool, at least in my opinion, with spatial data is that you can then transfer this information, since you know the spatial coordinates of each data point, from gene expression space to the actual spatial space. And that is what you see here in the rightmost picture. And that's just basically the spatial distribution of our clusters. Again, each point is colored by cluster identity. And if you now reference this with a annotation made by pathologists completely independent of our clustering results, you can see that the unsupervised clustering identifies similar morphological regions to what the pathologist spotted. For example, this orange region here, it was classified as an in situ cancer region. And very similarly here in the clustering results, we see a demarcated region, which we call it in orange just to make it comparable, of course. And what's even neater is that if you look at this part here, it's also annotated as in situ cancer, so it should be similar to the other orange region. And also in the clustering, we see that these three dots are assigned to the same spatial cluster, even though they are spatially disconnected. Speaking of trajectory inference, I said that there were basically two ways of doing this. I am not super fond of treating spatial data as single cell data.
Alma, your sound disappeared a bit now.
Ah, damn. When did you lose me?
Just slides.
Sorry?
Just when you switch to the next slide.
Okay, so I didn't have had the time to say too much then, but I'm back again, I guess.
Yeah, now it sounds good.
Great. Yeah, sorry for the bad connection. Yeah, like as for the trajectory inference here, there are mainly two ways of doing this. Like either you treat your data as single cell data, or you try to like modify your algorithms. And I'm not super fond of treating spatial data as single cell data because it's not. So here we see a nice example of when they actually try to repurpose one of these or reconstruct an already existing algorithm. And they applied a very similar model to what they used in the original velocity paper to merfish data. So if you remember, or perhaps have seen in the original paper by Alma et al., they used the ratio between unspliced and spliced mRNA, and then use this dynamical model to infer the position and pseudotime of certain cells. Very similar, but slightly different. Chia et al., I'm not super sure if that's correct pronunciation, but this group at least, looked at the ratio between nuclear mRNA and cytoplasmic mRNA, and then used an identical dynamic model here to infer pseudotime or like these transient cell states in their merfish data.